First of all, thanks for the invitation to come here. I'm very pleased to be able to introduce to you our company, Novo Biosciences. Just by way of introduction, Novo is a late-stage preclinical regenerative medicine company, and unlike the vast majority of other companies in this space that are focused on cell therapies, tissue engineering approaches, genetic approaches, et cetera, our approach has been to develop first-in-class small molecules that activate our intrinsic tissue and organ regenerative capacity. We've got two fundamental foundations that the company is built on. The first is that humans actually do indeed possess robust tissue and organ repair and regenerative capacity, but that this capacity is actually reduced and inactivated during development, postnatal development, and aging in most tissue types. And that's illustrated on this graphic here, which is it shows low and high regenerative capacity as a function of age versus tissue type. And what we know is, you heard a little bit about this earlier today, is that the fetus has really remarkable scar-free healing and regenerative abilities. Newborn infants, including mice and humans, have remarkable regenerative abilities. For example, a newborn mice will completely regenerate its heart, scar-free, after a variety of injuries. But once that mouse hits about 1 week of age, it rapidly loses that capacity. There are at least two papers that I'm aware of in the literature that have shown that young infants, a few months old, who've survived a severe myocardial infarction will actually regenerate their hearts, and by the time they're 4 or 5 years old, the heart's largely normal. Anybody in this audience has a heart attack and survives it, your heart's permanently damaged. You don't repair that damage. So we clearly have this capacity. It gets inactivated as we age. We do have tissues in our body that retain robust regenerative capacity. The lining of the gut is constantly replacing and regenerating itself. The skin regenerates about once a month. Blood cells constantly regenerate. The liver is probably the most complex organ we have that does a fair job of regeneration. Skeletal muscle regenerates somewhat in response to certain kinds of injuries. But then we do have tissues and organs like the heart, the brain, the spinal cord that are just lousy at it. And so again, the take-home message here is that the biological mechanisms, the genetic circuitry, is encoded in our genomes. And we believe that if we can understand that, you should be able to develop small molecules to reactivate it. And I'll show you proof of principle today. The second foundation is that tissue repair and regeneration requires coordinated activation and regulation of multiple physiological processes. There's been an intense, very narrow reductionist approach on stem cells in this field. What we've learned in the last 5, 6, 7 years is that repair and regeneration in response to both acute and chronic injury involves a very, very elegant series of highly coordinated physiological processes. Yes, it involves stem cell progenitor activation, but there's also very elaborate immune system response to an injury, metabolic changes, remodeling of the extracellular matrix, angiogenesis, so on and so forth. So there are many, many physiological processes that are important for repair and regeneration. They all have to work together to give rise to effective repair of a damaged tissue. And we believe that focusing on one of these alone in a very reduction approach is not going to get us to effective therapies. The approach we utilize is primarily an unbiased phenotype-based screening approach in whole animals. Typically, we are utilizing small molecules that already have some sort of toxicological profile behind them, so relatively safe up front. So we're looking at repurposed molecules. Once we identify these small molecules, we utilize them to develop a deeper systems-level understanding of all these processes that are important for regeneration. And by doing that, we've been able to identify a whole variety of new therapeutic targets. Through this small molecule approach, we've been able to inhibit fibrosis and activate a variety of these different processes over here, including modulating the immune system and how it responds to an injury. The proof of the principle here is our lead candidate molecule, which goes by the inelegant name of MSI-1436. It's also known as trodesquamine. I always have to think about how to pronounce that, so I'll just say MSI-1436. This was identified in a completely unbiased phenotype screen. It's a greatly de-risked molecule. It's already been through a phase 1 clinical trial for a completely unrelated indication about 10 or 12 years ago. And we've been able to show in diverse animal models that it's activating regeneration at doses 50 times lower than the maximum well-tolerated human dose. Our work's been extensively peer-reviewed, funded through peer-reviewed mechanisms. It's also undergone, for one of our clinical indications, a very successful pre-IND review with the FDA. So one of the things that we have focused on primarily at Novo is heart injury and heart disease. And I think everybody in this room knows that cardiovascular disease is really the leading cause of death in the world. And if you just look at ischemic heart disease alone in the U.S., it is a massive problem. It's about $130 billion a year problem, and it's the cost of treating ischemic heart disease is rising rapidly. The adult heart, as I said early on, does not regenerate after injury. And this is due, again, to the inactivation of those intrinsic regenerative processes during postnatal development and during aging. Over the last 10 years, there's been an intense focus on stem cell therapies, transplantation, stem cell transplants, in an effort to reduce ischemic heart injury and chronic heart injuries. All of those attempts have failed, and the field is very rapidly backing away from that now, despite really massive investment of both dollars and R&D effort. And to date, there are really no approved regenerative medicine therapies for heart injury. So this is one of the key areas that we focused on at Novo. What I'm going to show you here are some studies in mice where we used a very standard ischemic heart injury model. Again, everybody knows that a heart attack or a myocardial infarction is loss of blood flow to a portion of the heart. Those cells die, and in adults, they're basically replaced by dead scar tissue. And that's illustrated down here in the cartoon. The dead cells replaced by scar tissue. That causes further damage to the heart. You get dilation of the ventricles, and you lose the pumping capacity of the heart, ultimately leading to chronic heart failure. So what we did in these studies is we, again, used normal wild-type mice. We did a canonical injury where we permanently ligated the left anterior descending coronary artery. We waited 24 hours after surgery to make sure that there was indeed severe injury to the heart, which we confirmed by echo. And then we conducted all of our studies. In fact, everything we've done with this molecule has been done blinded and randomized. And we treated those mice either with vehicle or the drug for 4 weeks. We assessed function by echocardiography and also looked at the heart histologically. And what you see here are both functional measurements of heart injury as well as scar tissue formation. Over here, you're looking at echocardiography measurements of heart pumping capacity, either ejection fraction or fractional shortening. The black bar are normal, uninjured mice, so that's normal pumping capacity. The white bar are mice that are treated with vehicle only, and this is measured again 4 weeks after the initial injury. I will tell you that the 24-hour injury is no different from the 4-week injury. So right after you induce the injury, there's severe irreversible injury in normal vehicle-treated mice. But treating these animals with an extremely low concentration of MSI, 0.125 mg per kilogram, or 10-fold higher, we're seeing a 2 to 2.5-fold improvement in the pumping capacity of the heart. Over here, we see histological examination of the heart, cross-section through a vehicle and a drug-treated animal. These are 3 days after injury. They've actually only received 1 treatment of the drug by that point in time. This blue is the initial laying down of collagen that will eventually turn into scar tissue. We can quantify that, and what you can see is the scarring index in both the control and the drug-treated animal is about the same. We've got about 40% scarring that occurs right after the injury. 4 weeks later, this is what we see here. We have 2 different measurements of, again, the scarring index, length, and area. And what you can see is we're seeing a 50+% reduction of the infarct, the scar tissue, treating with MSI-1436. The important thing here is when you look at this graph and compare it to this one, the initial scar is 40% in both control and drug-treated animal. At the end of 4 weeks, it's 20%. So we're not simply blocking the formation of the scar, we're actually allowing that initial scar that's being laid down to resolve itself, to be reabsorbed and replaced, presumably with functional tissue. The other thing you can see over here when you compare these 2 groups, you see profoundly thinned ventricle wall in the vehicle-treated animal. That's again illustrated by the cartoon. This is because basically all the cells are dying into that scar. We never see that in the drug-treated group. Whenever we see scar tissue, it's always underlain by what appears to be normal functional muscle. So we're restoring heart function and we're reducing the scar with the molecule. We also did EDU labeling in these studies to look at cell cycle activity in cardiomyocytes. We looked at both a zone remote from the injury, from the infarct zone, and at the infarct zone. There's no measurable EDU incorporation into the DNA at a remote zone in either drug or a vehicle-treated animal. But you can see something quite different when you look at the infarct zone. Again, there's a few EDU labeled cells here in green in the vehicle treated, and obviously a massive difference when you treat with MSI 1436. If you look at high magnification and you co-stain with alpha-actinin, which is a contractile protein specific to cardiomyocytes, you can see here, I hope you can see that, but very beautifully, you have nuclei labeled with EDU in the cardiomyocytes. You can quantify that, and what we can see here is a 6-fold plus stimulation of cardiomyocyte cell cycle activity just at the infarct zone in the presence of MSI 1436. So we're reducing the injury, we're reducing infarct, we're improving function, and we believe this is due to regeneration of the heart tissue. This work was just published a little over 2 years ago. It actually received quite a bit of press coverage, and outside experts weighed in on what they were seeing. I'll just refer you to this one here. This is Nilesh Samani. He's a medical director of the British Heart Foundation. He said that it was surprising that a single molecule had such extensive and complete effect on the heart. And as best we can tell, MSI is the only small molecule that has ever been shown to activate regeneration in the adult mammalian heart. We've also looked at other diseases that cause heart injury, and this is partly because of scientific interest, but also business decisions. And the area that we focused on is Duchenne muscular dystrophy. Duchenne's is a fatal degenerative muscle disease. It affects all the muscles really in the body. It's caused by a variety of different kinds of mutations in the dystrophin gene. It's a rare or orphan disease, about 1 in 3,500 male births. Less than 13% of the patients who have this disease have treatment options other than steroids. Sort of amazing that 30 plus years after we understood the molecular basis of the disease, the best that we can really offer patients are steroid treatments. None of the existing, which is really one, Sarepta, the existing so-called genetic therapies have been shown to have any clinical efficacy. So really, best you can do for patients right now are steroids. Most of these kids lose the ability to walk and are confined to wheelchairs by the time they're 12 or 13, and most of them are going to die from either heart or respiratory failure because those muscles also degenerate. So what we did here in these studies is we took the D2B10 DMD mouse model, which is the model that is thought to most closely mimic the human condition. It's also a model that develops severe heart disease. Over time, as these animals age, they'll develop an enlarged heart and dilated cardiomyopathy, which is, again, what ultimately kills most of these patients. And what we did, again, in a 14-week blinded randomized study is we started the animals at 8 weeks before they had any real outward severe injury, treated them with MSI 1436, terminated the study at 14 weeks after the start, and then did histological analysis, et cetera. And what you can see is something quite striking. These images are both at the same scale here. As soon as we isolated the hearts from the vehicle-treated animal, you could see gross enlargement of the heart, about 30+% enlargement of the heart tissue. You can also see here in the area that's outlined in white, fibrosis. So again, beginning of severe fibrosis, which begins in the outside of the heart and proceeds inward. The heart from the MSI animals are completely normal size, no outward signs of any damage, any scar formation, et cetera. Cross-section through the hearts, again, confirm the size of the heart. Left ventricle, this is a major pumping chamber of the heart, is largely unscathed at this early stage of the disease. But what we're seeing is very severe injury to the right ventricle. You can see here that the wall of the ventricle is severely thinned in the vehicle-treated group, and it's normal here in the MSI-treated group. You can confirm that with echocardiography measurements. Again, the red bar is a wild-type mouse. That's a normal right ventricle wall thickness. Severe thinning shown by the blue bar in DMD mice that are treated with vehicle only. And the mice treated with MSI, we've basically restored normal ventricle wall thickness. Now, I want to be cautious about this next slide. I actually debated a lot showing these data because we do consider them early stage, but they're so tantalizing and I think so relevant to the group, I think they're important to show. In patients who have Duchenne muscular dystrophy, in young children, they don't show any outward clinically observable signs of heart damage. There are biomarkers that show they start to exhibit heart injury as young as 4 and 5 years old, but they really don't show severe heart injury until they're in their late teens, very early 20s. And that's what you see in the mice, too. At three months of age, the ejection fraction is roughly normal to wild type. But by the time these mice are 1 plus or minus years old, they have, again, what would be defined as severe defects in heart pumping capacity. We took these aged mice and we treated them with either vehicle or MSI-1436. We have 2 independent experiments so far. Cohort A, the green is vehicle only, and what you can see of the 2 animals in that experiment continued to worsen. 2 of the 3 animals treated with MSI showed remarkable 2 to 3-fold improvement in cardiac left ventricular ejection fraction. We did have 1 animal that continued to decline and actually died before we could complete the experiment. We have another group that's going now. We just got these data last week. Again, green is vehicle, blue is drug. And what you can see is 3 of the 4 vehicle-treated animals at 2 months are continuing to decline. 1 showed some improvement. 3 of the 4 animals that are treated with MSI are actually improving. Again, 1 quite strikingly, a 3-fold increase in cardiac ejection fraction. So what these data suggest to us is that not only can we treat an acute injury, not only can we slow the degenerative changes that are associated with a mutation in a gene like dystrophin, but we might also be able to reverse, or at least forestall, severe chronic injury in this particular animal model and possibly in other chronic heart injury situations as well. This slide basically shows that it works not only on cardiac muscle. This is skeletal muscle, and we're measuring it in 2 ways. Again, these are young mice treated for 14 weeks with vehicle or MSI. The black is MSI, open is vehicle, and this is a measurement of grip strength. It drops, but typical for this animal model in the vehicle-treated group, and MSI preserves normal grip strength function of the skeletal muscle. And then over here we have histology. This is very sick muscle, loss of the bundles, muscle bundles, the fibers, infiltration of scar tissue, immune, fatty deposits, etc. And the tissue in the MSI group looks largely normal. So how does this stuff work? MSI is a natural aminosterol compound. It's a potent inhibitor of protein tyrosine phosphatase 1B. And unlike the other 300 or so inhibitors that humans have developed, MSI acts via a very unique mechanism. PTB is a ubiquitous enzyme. It's expressed in every cell type and tissue as far as we can tell. And its primary role in life is to inhibit receptor tyrosine kinase signaling. And it does that very indiscriminately. It doesn't matter whether you're an RTK for insulin, an RTK for a specific cytokine, or a growth factor. PTB doesn't care. It basically dephosphorylates and inactivates the activated receptor, and thus inhibiting all the downstream signaling processes. What's becoming very clear as we start to understand these integrated physiological processes that are important for repair and regeneration is that many of them are controlled by RTK signaling. And in fact, I would put my neck out a little bit and say when we fully understand these, we're a long ways from that, is that RTK will probably be the predominant signaling processes that are involved in regulating these. So what you have here is PTB1B basically shutting these processes off. And very simply what MSI is doing is it's inhibiting PTB1B, allowing these endogenous signaling processes, the RTK signaling processes, to remain active, possibly amplifying them, and allowing all these physiological processes to remain active and allowing them to in turn stimulate the repair processes. We have a lot of data to support this. Very large body of data. I'll show you one experiment here. What we did is we induced an ischemic injury in a PTB1B knockout mouse. Those mice have been around now for about 2 decades and been studied quite extensively. And what you see here in the inset are data I've already shown you. Normal ejection fraction in an uninjured mouse. Injured mouse 4 weeks after injury, treated with MSI, again, 2.5-fold repair. Over here, we're looking at the knockout mouse. The black are mice, PTB1B knockout mice treated with vehicle only. Again, you have severe injury, you have recovery. If we treat with MSI, there's no additive effect. You're seeing the same recovery. So we can phenocopy the effect of the drug with PTB1B knockout, and the knockout plus MSI, there's no additive effect. We have the same results in another animal model, lots of gene expression data. There's a lot of papers in the literature that show that PTB1B knockout mice have improved repair and regenerative abilities. They also have slowed cardiovascular degeneration with aging. Just to finish up, summary, Novo is a late-stage preclinical regenerative medicine company. We're looking for small molecules that activate our intrinsic capacity to regenerate. And our lead candidate, as I've shown you, activates repair and regeneration of heart tissue and skeletal muscle in mice, also other animal models we've looked at. And we've currently got a lot of success with peer-reviewed work, peer-reviewed funding, investor funding, et cetera, and a successful pre-IND review. Just to wrap up, I want to acknowledge the key people doing the work. Vut Yin is the Chief Scientific Officer for Novo. He's done all the hands-on work together with Ashley Smith and a PhD scientist, Adrian Rodriguez. So I'll stop there, and again, thank you for the invitation. Happy to take questions.